Hello everyone, I am Dr. Suresh, Faculty of Biochemistry, and in this short video we will be covering of complexes of electron transport chain and its inhibitors. So first of all, what is electron transport chain? Electron transport chain is a mechanism in transferring reducing equivalents and electrons across the complexes to generate ATP. From where these reducing equivalents are generating? From the various metabolic pathways like carbohydrate metabolism, and also you say lipid metabolism, protein metabolism, nucleic acid metabolism. So, and all these metabolisms are involved in generation of these reducing equivalents, which will pass on through one after the other these electron transport chain complexes and finally to generate ATP. Right. So, totally before going in details, first we will see where is the location for this electron transport chain. Mitochondria, but what I want you people to remember, yes, mitochondria is the organelle where electron transport take place, but what is exact location? Inner membrane, okay, because mitochondria is having two membranes, one is outer membrane, other one is inner membrane. So, this electron transport chain specifically takes place in inner membrane of mitochondria. That is the thing I would like to remember all of you. And the next is complexes. How many complexes this ETC is having? So, there are total of 5 complexes. They are like complex 1, complex 2, complex 3, complex 4 and complex 5. So, here it again you can say with Roman numbers 1, 2, 3, 4 and 5. So, the other names of these complexes we will discuss now. Complex 1 otherwise known as NADH dehydrogenase complex. What is that? NADH dehydrogenase complex, okay, and complex 2 otherwise known as succinate dehydrogenase complex, and complex 3 is known as cytochrome reductase complex, cytochrome reductase complex, and complex 4 is known as cytochrome oxidase, cytochrome oxidase, whereas complex 5 is known as F0 F1 particle or ATP synthase, okay, ATP synthase. So, these are all the complexes involved in ETC. One is complex 1 is NADH dehydrogenase complex, complex 2 is succinate dehydrogenase complex, complex 3 is cytochrome reductase complex, complex 4 is cytochrome oxidase complex, and complex 5 is F0 F1 particle or ATP synthase complex. So, now we will have quick review on inhibitors. So, now as we have seen the things here, 5 complexes are there. There are different inhibitors do exist for these complexes. So, first to talk about complex 1, what are the inhibitors? So, first thing is phenobarbital and rotenone. So, 1, 1. So, you see the last 1, 1, phenobarbital like barbiturates, phenobarbital 1 is there in the last, rotenone there is 1 in the last, and we are inhibiting 1. Okay. So, complex 1 inhibited by 1, 1, 1, like that you can say 1, 1 you can remember. Okay. Complex 1 inhibited by phenobarbital and rotenone, and complex 2 if you are talking about by malonate, because it is structurally like what to say, malonate concentration, okay, inhibits the succinate dehydrogenase enzyme, and cytochrome reductase inhibited by antimycin otherwise British plebiscite antigen, BLA, Okay, sorry, it is not BLA, it is BAL, British anti-lewisite agent, BAL, British anti-lewisite agent, okay, BAL. And complex 4, it is cytochrome oxidase. It is the target question for competitive examinations. Remember, cytochrome oxidase inhibited by carbon monoxide, cyanide and hydrogen sulfate. Okay, so these are the three things which inhibit complex 4. So remember, directly they will not ask in competitive exams. They will ask like what? So cytochrome oxidase inhibited by the following. They will give multiple options. So if any one of the option is having carbon monoxide or cyanide or hydrogen sulfate, you have to pick that, right? And complex 5, it is inhibited by oligomycin. Right, in this quick table we have seen complexes, other names of these complexes and the inhibitors. And one more point you have to note down here, uncouplers. What do you mean by uncouplers? So we have learned that electron transport chain is oxidative phosphorylation. In 4 complexes oxidation is taking place, okay, and in the 5th complex phosphorylation is taking place where ADP is phosphorylated to form ATP, right. So here ATP is at one part and oxidation is one part, so both are combined. So that's why electron transport chain is known as oxidative phosphorylation. Got the point? So here there are some uncouplers which separate this oxidation and ATP synthesis part. So they are known as uncouplers. They are uncoupling the oxidative phosphorylation. Okay, they may allow the oxidation but they will not allow the ATP production. So that is a significant point about uncouplers. So there are 2 types of uncouplers are there, chemical and biological. So chemical couplers you can talk about like 2,4-dinitrophenol and aspirin. So these are the 2 chemical uncouplers. And if you are talking about biological uncouplers, they are otherwise known as physiological uncouplers. They are naturally produced in our body. So dermogenin is the one, is a protein, is a substance which present in brown adipose tissues of infants. Okay. So that's why baby's temperature, if you see, they are like always like high in temperature, okay, because of this brown adipose tissue, because instead of ATP production most of the energy dissipated as heat, okay. And the next thing is thyroxine, free fatty acids and bilirubin. So they are all acting as natural or biological uncouplers. They will allow the oxidation part, okay, but transferring of reducing equivalents from one complex to other complex, finally the oxidation of oxygen and reducing equivalents to water, but not the ATP generation. So that is a significant one. So that's all about the complexes of electron transport chain and its inhibitors. For competitive exam point of view, this review may help. Thank you.